Hello, everybody. So I'm going to be talking about a new field, not a new field, but an emerging field called as Metaproteomics, which is kind of an extension of metagenomics. And we had at least 3 talks on the first day on using metagenomics approaches. So I'm going to be talking a little bit about that. The title actually is a wordplay on not only the diversity of microorganisms that are present in a microbiome, but also the diversity of tools and the complexity of the tools that we use for these analysis. What I'm hoping to do, again inspired by the talk about hackathon by Beyond, is I'm hoping that I can convince you that this is an interesting field enough so that we could have a hackathon at some point. So just to kind of introduce the field, microbiome is basically the DNA that's present not only in your, actually the DNA that's present in microorganisms in your body. It's an extremely interesting field because the gut microbiome is one of the most studied system right now in research. And the reason for that is because there have been studies that have implicated microbial flora to have effect on health. And there are quite a few studies, diseases that have been mentioned here. And the idea is to find out what's the composition of these microbiomes in your body and how does it affect your physiology. So metagenomics is one of the tools that is used, especially when you're using DNA sequences. But what metagenomics ends up doing is it ends up giving you a taxonomy of the bacteria that are present. It does not necessarily give you an idea about what are the proteins or what are the functions of these bacteria or how are these bacteria actually affecting your body. So for that, there are either 2 methods one can use. One method is metatranscriptomics, wherein you actually look at the RNA translation, the RNA expression of these microorganisms, or proteomics, which is the area that I come from. And this particular field was introduced way back in 2004. One of the interesting things that has come out after 10 years of research in this field has been that it's not necessarily important that it's not necessarily important what the taxonomy of your microbiome is, but one should look at what's the function of this microbiome. So, for example, if you were to have microorganisms of similar taxonomy as has been shown here, and if they were subjected to dissimilar conditions, you would get a response or function or expression of proteins which is different than what your taxonomy suggests. You could also have another scenario wherein you could have organisms which are of dissimilar taxonomy, so composition that's entirely different. It could be subjected to same conditions, and hence you could actually end up getting similar proteins or genes that are getting expressed. So one other thing that has come out just two months ago, the White House actually came out with a paper suggesting that researchers should start utilizing resources that are available. They actually launched an initiative called as National Microbiome Initiative, and the idea here was to bring in researchers from various fields together on a common platform so that one can look deeper into microbiome studies. In 2015, last year, we actually published a basic blueprint of doing metaproteomic analysis. So there are quite a few tools available here. I won't go into details of this, except to say that we have actually used these for various studies, either in dental caries for looking at cervical-vaginal fluid microbiome or lung microbiome. What I'd like to talk about is take this blueprint and try to add newer tools into this or newer workflows into this. And again, I don't have time to go into the details of these, but there are multiple avenues wherein we could actually increase the number of tools or workflows in all of these various topics that I've mentioned. And what I'd like to talk about is, we would actually like to take this basic workflow, but also maybe consult researchers from all around the world who are metaproteomics experts, identify tools and workflows, and then once we prioritize these tools and workflows, we would like to approach the Intergalactic Utilities Commission, and then hopefully we could have a really focused hackathon based on the ideas that come out. And eventually then we would like to test these workflows and have them available to researchers so that the field of metaproteomics can flourish. If there are any questions or suggestions, I'll be very happy to take them. The last slide is just basically an acknowledgement that this is a work of the Galaxy P team, wherein multiple researchers, collaborators, developers have been involved in making this possible.
Thank you, Prateek. Do we have any questions for Prateek? You sort of anticipated almost all of mine. When you started out, I was going to ask about the National Microbiome Initiative, and you had a slide about that. I was going to ask about a role for Galaxy in relation to that. You had a slide about that. Asking about new tools. So I guess as far as timeline for aligning with that national initiative, what do you see as next steps? Presumably there are things to do before Galaxy 2017.
I think the timeline is, I think, next 2 or 3 years. These, whatever, $120 million from various federal agencies is going to be available for not only metaproteomics, but also metagenomics and, microbiome research as such. But in terms of Galaxy, I think, for metagenomics, there are already tools available. There definitely is at least in the coming from a proteomics background, there's definitely an interest, and it is also an emerging area. So I think within next year, we should be able to put in tools and workflows in place so that they could be used. Yeah, thanks.
Great. I'd like